Hello and welcome to Sam Biology class. Friends, today we shall discuss about loops, turns and bends that play an important role in protein structure and function. Roughly half of the amino acid residues in a typical globular protein reside in alpha helices and beta sheets, remaining half in loops, turns, bends and other extended conformational features. Protein loops are the patternless regions that connect two regular secondary structures. They are not well defined. Despite the lack of pattern, loops are not completely random structures. They contain hydrophilic amino acid residues and are located on protein surface. Loops and turns connect adjacent strands in a beta sheet and also adjacent alpha helices. Loops having 4 or 5 amino acids are called turns or bends. So turns and bends refer to the short segment of amino acids that connect two units of secondary structure, for example, two adjacent strands in a beta sheet. Turns have regular hydrogen bonding pattern and specific dihedral angles. In a globular protein which is roughly spherical, by turns and bends, the polypeptide chain changes its overall direction repeatedly. As a result, it folds upon itself, thereby forming a compact structure. Based on the conformation, turns can be classified into several classes such as alpha turn, beta turn, gamma turn, delta turn and pi turn. First of all, what is alpha turn? An alpha turn is a tight turn involving 5 amino acids. The amino acids are designated as I plus 1, I plus 2, I plus 3 and I plus 4. So there are 5 amino acids in an alpha turn. It is stabilized by a hydrogen bond between the carbonyl oxygen of first amino acid and the amide hydrogen of fifth. This is the hydrogen bond which connects the carbonyl oxygen of first amino acid and the amide hydrogen of fifth, as a result of which this turn becomes stable. At alpha turn, a polypeptide chain reverses its overall direction, as a result of which it folds on itself by 180 degrees. In an alpha turn, the end residues are separated by 4 peptide bonds. Based on the backbone dihedral angles, that is phi and psi of residues I plus 1, I plus 2 and I plus 3, alpha turns are divided into 9 different groups. 9 different groups such as 1 alpha LS and 2 alpha LS, 1 alpha RS, 2 alpha RS, and the remaining varieties are 1 alpha LU, 2 alpha LU and 1 alpha RU and 2 alpha RU and 1 alpha C. So these are the 9 different types of alpha turns, all involving 5 amino acids and the distance between the alpha carbon atom of I and I plus 4 residue is less than 7 angstrom. Alpha turns play an important role in the structure and function of proteins. They are characterized by the presence of hydrophilic amino acids. The structures are not only exposed to solvent but also protrude from the protein surface as hook-like shape that interact with various other components. So friends, it is because of alpha turn a polypeptide chain reverses its direction and folds on itself to form a compact structure which is seen in many globular proteins. Turns are non-repetitive secondary structures. Beta turns are one of the most common structural motifs in proteins. It is also known as tight turn, reverse turn, beta band and Venkatachalam turns. A beta turn is a 180-degree turn involving 4 amino acids which are labeled as I plus 1, I plus 2 and I plus 3. Each beta turn is stabilized by an inter-residue hydrogen bond which connects the carbonyl oxygen of first and the amide hydrogen of fourth amino acid. So this is the inter-residue hydrogen bond which provides stability to the beta turn. In a beta turn, the end residues are separated by 3 peptide bonds and the peptide groups of central 2 residues do not participate in hydrogen bonding, and the distance between the alpha carbon of residue 1 and that of 4 is less than 7 angstrom. At a beta turn, the polypeptide chain changes its overall direction by 180 degrees, and as a result the peptide chain folds upon itself to form a compact structure that we see in globular proteins. Beta turns connect adjacent strands in an antiparallel beta pleated sheet. Hence it is named as beta turn. Beta turn is a tight turn which reverses the direction of the polypeptide chain. Hence it is also called as reverse turn. It is a short segment of 4 amino acids that produce a band in the polypeptide chain. For example, a polypeptide has one such short segment of 4 amino acids. So during folding, the polypeptide chain will form one beta band in this region which contains these 4 specific amino acids. So this is the beta band which is stabilized by the formation of a hydrogen bond between the carbonyl oxygen of one and the amide hydrogen of another amino acid. So this is the beta turn, and if the polypeptide chain has several such short segments of amino acids, then it will fold upon itself several times, thereby forming a number of beta turn that will make the region more stable. So in this way, beta turns connect adjacent strands in an antiparallel beta pleated sheet. Based on the torsional angles phi and psi of residue I plus 1 and I plus 2, beta turns are classified into 4 different groups such as type 1, type 1 dash, type 2 and type 2 dash. These are the values of phi and psi which favor the formation of different types of beta turns. Type 1 and type 2 beta turns are very common. They differ mainly in the orientation of the amide bond between residues I plus 1 and I plus 2 on the plane of the beta tone. The other two groups, type 1 dash and type 2 dash, are the mirror images of the backbone conformation of type 1 and type 2 beta tones respectively. In type 2 beta tone, the oxygen atom of the second residue crowds the beta carbon atom of residue 3, which is usually glycine. Whereas in either type of beta tones, type 1 and type 2, second residue is often proline. This is because it can assume the required conformation. Statistical analysis reveals that glycine and proline are more favored in beta tone, presumably because of the flexibility of glycine and because of the cyclic structure of proline. In addition to this, hydrophilic amino acids such as asparagine and aspartic acid show a higher propensity for the formation of beta tone. This is because they are present on the solvent exposed surface of the protein. In addition to their role in protein folding, beta tones also serve as recognition motif for protein-protein interactions. Gamma tone is a tight tone. It consists of three amino acid residues which are labeled I plus 1 and I plus 2. In a gamma tone, and residues are I and I plus 2, and the middle residue is I plus 1. Such tones are stabilized by an hydrogen bond which connect the carbonyl oxygen of one amino acid and the amide hydrogen of another amino acid, which is two residues ahead in the polypeptide chain. So the hydrogen bond is formed between residue I and I plus 2. So residue I is hydrogen bonded with residue I plus 2. In gamma tones, and residues are separated by two peptide bonds. This is the inter-residue hydrogen bond which connects residue I and I plus 2. This bond provides stability to the gamma tone. This figure shows a part of the polypeptide chain, and here you can see that this is a short segment of three amino acids which constitute one gamma tone, and the inter-residue hydrogen bond is formed between the carbonyl oxygen of one amino acid and the amide hydrogen of another amino acid, which is two residues ahead in the sequence. So gamma tones are short segments of three amino acids stabilized by an inter-residue hydrogen bond. Gamma tones are fairly common in proteins. They are usually found in the ligand binding site or the active site. On the basis of dihedral angles phi and psi of the middle residue, gamma tones are classified into two different groups such as classic gamma tone and inverse gamma tone. Inverse gamma tone. Classic gamma tones are far less common in proteins. They are usually found at the loop ends of beta hairpin. Except one, all are associated with the reversal in the direction of the polypeptide chain. One classic gamma tone occurs at the edge of the second hypervariable region of the light chain in some immunoglobulins. For crossing gamma turn, phi is equal to 70 and psi is equal to minus 60. Second type of gamma turn is inverse turn, for which phi is equal to minus 70 and psi is equal to 60. Inverse gamma turns are more common in proteins. Usually they are not associated with the reversal in the direction of the polypeptide chain. Such turns give rise to kinks in the polypeptide. They are considered as an intermediate in folding and therefore stabilize in strands before the formation of beta-pleated sheets. Delta turn is the smallest type turn. It consists of two amino acids which are labeled I and I plus 1. Such turns are stabilized by an intraturn hydrogen bond which is formed between the carbonyl group of one amino acid and the amide group of another amino acid which is located next in the polypeptide chain. So the hydrogen bond is formed between residue I and I plus 1. In delta turn, the two residues are separated by one peptide bond. This figure shows the part of a polypeptide chain. These are the constituent amino acids which are connected by peptide bonds. Here, this is the delta turn which consists of two amino acids, and this is the hydrogen bond which connect the carbonyl group of residue I and the amide group of residue I plus 1. Like other turns, delta turn is also located on protein surface, and in doing so it contributes to the protein structure and function. Like other type turns, delta turn also changes the overall direction of the polypeptide chain that help in protein folding. Another type turn is pi turn. It is the largest type turn which consists of 6 amino acids which are labeled I plus 1, I plus 2, I plus 3, I plus 4 and I plus 5. Like other type turns, pi turn is also stabilized by an inter-residue hydrogen bond, stabilized by an inter-residue hydrogen bond which connect the carbonyl group of one amino acid and the amide group of another amino acid which is located 6 residues ahead in the sequence. So the inter-residue hydrogen bond is formed between amino acid I and I plus 5. This hydrogen bond provides stability to the turn. In pi turn, the end residues are separated by 5 peptide bonds. Because it changes the overall direction of the polypeptide chain, pi turn is also involved in protein folding. So friends, this is all about loops, turns and bends. For more updates, please subscribe my YouTube channel Sam Biology Class.